The first session will be co-chaired by Professor Hidenori Arai, who is the president of National Center for Geriatrics and Gerontology in Japan, and by Professor Lin Qihong, who is the president of National Yangming Jiaotong University.
Okay, so good afternoon. My pleasure to moderating with my old friend, Professor Arai, for this session. The first speaker this afternoon is by Professor Ting-Fen Tsai, Tsai Ting-Fen, and basically says okay, so Professor Tsai is currently the distinguished professor in previously National Yangming University and right now the National Yangming Jiaotong University. She's world-renowned. I don't know you want to call yourself gerontologist or a cell biologist still, okay? And she's gonna give us a very interesting talk about rejuvenations, turning back time by enhancing 62 prolongevity gene. So let's welcome Professor Tsai.
Thank you for the introduction from President Lin. Today I'm going to talk about introduce the E2 prolongevity gene and update the progress in this longevity genes, and for all the friends, including the friends from Japan online and Toba. The CDGSH ion sulfur domain are in short CISD protein families, and currently there are three families. There are three genes in the families, including the 61, 62, and 63. 61 is located in the mitochondrial outer membrane. However, 62, it has a very similar structure with the 61, have transmembrane domain, and the ion sulfur binding CDJS ion sulfur binding domain is located in the mitochondrial outer membrane, the ER, and the mitochondrial associated ER membranes. For the human 62 homodimers, each monomers that contain 2 ion and 2 sulfur clusters. And this ion sulfur cluster traditionally is responsible to accept and release the electrons. Based on the human aging genomic resources, currently there are 8 prolongevity genes in the mammals. And the 62 is one of the prolongevity genes in mammals, and this actually is based on majority the study published from my lab. Regarding the function of 62, 62 actually regulates the calcium homeostasis and the mitochondrial functions. And for example, in the hepatocytes, 62 will interact with the SERCA2B, and SERCA is a calcium channels that will pump the calcium from the cytosolic into the ERs, so to maintain the calcium homeostasis. And the 62 also located in the mitochondrial outer membranes, and to maintain the mitochondrial function. So during haploinsufficient 62, and this will lead to increase of the oxidative modification and decrease of the calcium channel activity of the SERCA. So this will cause the increase of cytosolic calciums and leading to the mitochondrial calcium overload and the dysfunction. And this increased cytosolic calcium also will turn on the ER stress and lead to fatty liver development in the liver. So 62 deficiency will cause a variety of age-related disease. For example, that will cause the corneal degenerations, sarcopenias, heart failures, arrhythmias, as well as the fatty liver development just mentioned, and the adipose tissue atrophy. In addition, the 62 deficiency will accelerate the A-beta mediated neuronal loss. And interesting, during the natural aging of wild-type mice, the 62 expression level is down-regulated. For example, in the muscles, at the middle age, at 13 months old, the 62 decreased to about 30% compared to the young mice at 3 months. And at the very old age, about 26 months old, the expression level is less than 20% in the old age. compared to the young age. And similar observation was observed, for example, in the heart and other organs. So indeed, the 62 knockout mice is an accelerating aging mice. In contrast, a persistent high level of 62 expression in the 62 transgenic mice, actually this mice is a very long-lived mouse. For example, look at the right mouse. This 62 transgenic mouse is at 34 months old. This equivalent in the human is about 105 years old. It looks younger compared to the wild-type mice at 15 months old. So this is the survival curve for the mice. So the blue line is the 62 knockout mice. The 62 expression level is 0. And the gray line is the heterologous mice. This is 50% of the 62. And the open box is the wild-type mice. It's 100% of 62 expression. The green line is the transgenic mice. They have 200% of 62 level. So based on this survival curve, obviously we have demonstrated there is a 62 dose-dependent increase of survival rate. So in the mice, the level of 62 decreased during natural aging. If we can maintain a consistent high level of 62 expression, then we can extend a healthy lifespan. So our purpose is to develop an anti-aging drug using the 62 as a molecular target. So we have generated 62 reporters to monitor the transcriptional activation of 62. We use a human 62 bacterial artificial chromosome. This bag contains more than 100 kilobase of the DNA that contain the entire 62 genes, as well as the upstream and the downstream sequence. So we knock in the luciferase, this reporter gene, into the exon 2 of the gene to establish the 62 reporter. So this is the 62 reporter mice, okay? And under specific treatment, the increase of the luciferase actually indicate the activation of the 62 gene. And this specific treatment, actually, I tell you the secret, is the starvation. So starvation will activate the 62 longevity genes. And we also established two 62 reporter cell platforms. The first one is the mouse embryonic fibroblasts established from the transgenic mice. The second one is the HEC293 that contains the 62 back reporter reporter cells. So regarding the compound libraries, we established a compound libraries from the noble herb that described in the classic of herbal medicine in Chinese, 神农本草经. And actually, this is a small compound libraries that's established only from the 60 noble herbs. And this compound libraries contain only 780 samples. But actually, there is a lot of the 62 activation inside. So this is a candidate list of 62 activators. For each of the 62 activators, we give a code name called PenZhu, in short, PZ. So who is the PenZhu? PenZhu actually is a very long-lived celestial beings in Chinese myths. So that indicate they have a very healthy aging and longevity. So PC19 is the compound I'm going to talk about. So PC19 is a pure compound. The purity is more than 95%. And it is a very potent 62 activator that can activate or stimulate 62 expression in both the 62 reporter cell and the reporter transgenic mice. So we provide the PZ, these compounds in the food to treat the mouse for a long time. And in the mouse, the dose we use is 100 milligram per kilogram per day. This equivalent in the humans is about 490 milligram per 60 kilogram per day. So definitely this is a dose that achievable in the human. This is the protocol for the late life treatment of PC19 in the naturally aged mice. We treat a very old mice, okay, at the age of about 21 to 22 months old. So this equivalent in the humans is about more than 17 years old. We treat the mouse for 6 months and obtain sacrifice the mouse and obtain the tissues at around 27 to 28 months old. So this equivalent in the human is more than 80 years old. So interesting, we got very exciting result. For panel B, this is the Western blot analysis for the level of 62. So we, after quantification, you see, for example, in the skeletal muscles, the 62 level downregulate to about 30% at old age. However, PC19 treatment for 6 months, they can significantly increase the 62, the longevity genes, to a level that is comparable to the young mice. And similar observation was observed in other organs, for example, in the heart. And most strikingly, we found they increase the median lifespan, and in particular, this is a healthy lifespan. So the blue line is the mouse treated with the PC, started at 21 months old. And the dark lines is the vehicle control. Gray line is untreated. So this data demonstrate that PC treatment can increase the median lifespan for about 9%. So this equivalent in the humans in Taiwan, for examples, the male will increase the median lifespan from 78 years old, plus 9% is increased to 85 years old. In the humans, it's increased from 84 years old to more than 90 years old. So I think this is quite impressive, at least to me. And this is monitor the whole body energy metabolism. So this data here is, we monitor the daytime and the nighttime. And for the mouse, they are active during the night times. And the green line is the control from the young mice at 3 months old. Blue line is the old mice. Okay, so, and the red one is the old mice treated with the PC19. So this data actually demonstrate that the PC19 can increase the whole body energy metabolism in terms of oxygen consumption, and similar observation for the CO2 production and energy expenditures. And most strikingly, we monitor the same group of mice, okay, when they are different at different age. So again, the green line is the young mice control, and the mouse, the orange line is the mouse at 23 months old. Dark line is at 28 months. So this data indicate there is an age-dependent decrease of the whole body energy metabolism. However, when we treat the PC19, you look at the dark line, this one is at 28 months old. The light blue is 23 months old. So this data actually indicate the mouse is getting older, but the whole body energy metabolism is getting better. So this data actually is demonstrated that PC19 appear to rejuvenate the aged mice in terms of whole body energy metabolism. We also apply the micro-CT to monitor the whole body fat and body limb. So actually these data demonstrate that the PC19 treatment that can reduce the fat accumulation. At the same time, they can increase the body limb compared to the vehicle control. And this is monitor the glucose homeostasis. And regarding the body weight, there is no difference for the body weight with or without PC19 treatment. And interesting, for the blood glucose, after the fastings, the blood glucose level is significantly reduced after the PC19 treatment. So actually, during the natural aging, there is a panel of differentially expressed genes associated with glucose metabolism. They are actually upregulated during the natural aging. So this probably explains why the basal blood glucose is upregulated during aging. However, after PC19 treatments, these differential expression, they are downregulated. So this is a summary to summarize that the PC19 actually slow down or even rejuvenate the heart ages in the naturally aged mouse. So if the young mice in the heart, the 62 level is 100%. When they're getting old, at 24 months old, the level of 62 downregulate to less than 50%. And for this transgenic 62 transgenic mouse, this is a long-lived mouse models. And the expression level at the old age is 100 to 30%. Very interesting, the old mouse treated with PC19s, the 62 levels that can upregulate to about 130%. So this level is similar to the long-lived transgenic mice. Okay, so for the monitor of cardiac functions, we found that the 62 activated PC19 that indeed can improve the aging-related dysfunction. Regarding the structure, the PC19 can improve the structural damage and the mitochondrial functions. And this is a summary for the skeletal muscle aging. And very similar, we found the PC19 can slow down the skeletal muscle aging. So this is the RNA sequence analysis. And it looks like quite busy, but we can just look at the colors. For panel A, the left one, this blue one is the panel differential express gene expressed in the young mice. The second is the line is the old red one. That means in the old age, in the heart, they are upregulated. However, these old mice, after PC19 treatment for 6 months, the differential express genes are downregulated. Okay, and we observe this in the heart and similar observation in the skeletal muscle. So these RNA sequence data actually demonstrate the PC19 that can indeed bring about a younger transcriptome patterns of gene expression during the old age. So one important question is that does PC19 delay the agents that is function in a 62 dependent manners? So to answer this question, we knock out the 62 genes in the muscles. We look at this blue box, okay? In the absence of 62, actually when we treat the PC19 totally, the beneficial effect is disappears. So this data actually indicate the PC19 delay the skeletal muscle aging indeed is in a 62 dependence. So this is another evidence from the RNA sequence. From the RNA sequence data, we know that about 80% of the differential express gene, the pattern will disappear once the 62 is absent. However, there are about 20% of the differential express gene that maintain their patterns even in the absence of 62. So that means 20% of the differential express gene, they are 62 independent. So but majority of differential express gene, they are dependent on the presence of 62. So the first part regarding the healthy aging and longevity, the conclusion is the PC19 that can promote the longevity and delay aging via activation of the 62 in the naturally aged mice. So the second part I'm going to talk about is the 62 activator and the skin agents. And this part is still unpublished data, so they are manuscript under the revisions. For the humans epidermis, most of the cell, 90% of the cell, actually they are keratinocyte, okay? And regarding the epidermis layers, okay, from the buttons, these are the basal layers, and then spinous layer, granular layers, and the conified layers. The basal layers contain most of the keratinocyte. They are proliferating keratinocyte. And they have a marker K14. This is for the basal layer. And this is the immunofluorescence staining of the 62 and the K14s using the human skin samples. So this data clearly demonstrates that the 62 actually is mainly expressed in the proliferating keratinocyte of epidermal basal layer in the human skins. If we obtain the skin tissue from the sun-exposed and the non-sun-exposed regions, the sun-exposed region, for example, the face and the neck, the non-sun-exposed region, for example, from the chest, okay? And this immunostaining showing very interesting data is the 62 actually they are downregulated in the sun-exposed size compared with the non-exposed size. The 62 always downregulate in the sun-exposed tissue of the skin. So one important question here is the PC19, they can improve UVB-induced skin photoaging. And if this is a function in a 62-dependent manner. So we use the wild-type mice and the knockout mice at 3 months old. We pre-treat the mice for PC19 for 7 days. After 7 days, we challenge the mouse with UVB irradiation once a day for a sequential 5 days. And after 2 days later, we sacrifice the mouse and examine their skins. So interesting, panel B is a Western blot analysis. Obviously, you can see the UVB irradiation will downregulate the 62 levels, and to about the quantification shows that downregulate to about 50%. However, with the presence of the PC19, the 62 level obviously is upregulated to a level actually there is seems like is stronger than the untreated the skins. And the other marker is MMP1. MMP1 actually is a markers major indicator for the UVB-induced damage in the keratinocyte. So this data clearly demonstrate that PC19 treatments that can significantly reduce the MMP1 that indicate that reduce the damage in the keratinocyte. So this is the dorsal skin from the mouse. Okay, after UV irradiations, what we see is the skin has a redness and they have a thick wrinkles. However, with the PC19 treatments, look at this skin. They look smooth and unwrinkled. Interesting, in the 62 knockout mice, this protection to the skins disappears. And because after PC19 treatment in the 62 knockout mice, the skin still look reddish. So this data clearly demonstrates that the PC19 indeed can ameliorate the UVB-induced skin damage and function in a 62-dependent manners. This is the histology staining with the Mason trichomes. So the mouse skin without any treatments, this is the mouse, the histology. After UVB irradiations, you see the skin, we see an increase of skin thickness. And this increase of skin thickness including the epidermis and the dermis. However, with the PC19 treatments, you see the thickness of skin is significantly decreased. So the next question is if that can rejuvenate or delay the skin aging. So this again is the same protocol we apply to see the healthy longevities. And this time we look at the skin ages. So interesting. Similar to the skeletal muscle and heart. Here, what we see is the level of 62 decrease during the aging, that decreased to about 50% at the middle age, and decreased to less than 30% at old age. And interesting, in the old mice, treated for PC19 for 6 months, that can significantly increase the 62 in the skins. And this increase to a levels that is similar, comparable to the young mice without any treatment. So this is a very amazing data. So we look at the histology of the skin. Panel at this one. This is the skin histology from a very young mice at 3 months old. You see a lot of hair follicle on the inside. Probably you cannot see, but there is the spatial gland. Spatial gland is the lipid-producing structures. And then the bottom one is the old mice, the skin from old mice. Obviously, you can see the degenerate hair follicle and degenerate of the spatial glands. However, after 6 months treatments, this is the skin. What did you see? What you see is the increase of the hair follicles. And also, sometimes we see a hair follicles associated with 2 spatial glands. So this data is obviously demonstrate the PC19 appear to rejuvenate the naturally aged skins in the old mice. We quantitate including the hair follicle density, the nucleus of the spatial gland, or the size of spatial glands. All of this data actually demonstrate that PC19 not only slows down the skin aging, but also rejuvenate the naturally aged skin. So I'm going to tell you the secrets. What is the PC19? PC19 has two structural analogs. One is called PC19A, and one is called PC19B. PC19A equivalent to hesperidin D, okay? The other one is hesperidin, is PC19B. And the PC19A hesperidin is present in the peel of the citrus fruit. So I think whenever you eat the oranges, usually throw away the peel. And the secret for longevity actually is in the peel. So remember next time, don't throw away the peel of the orange. Okay. So, hesperidin is we call PC19A. This structure has two sugars. And because of these two sugar residues, so it is not easy to be absorbed by the intestine. However, PC19A, the hesperidin, can be converted to hesperidin, the PC19B. And so move out. The sugar will be deleted, removed. And this PC19B, hesperidin, actually is the very potent SIS-D2 activators. So this is what just mentions. All of the beneficial effect is come from the SIS-D2 activator, hesperidin, okay? And some of the probiotics include the lactobacillus, or include the, and also the bifidobacteria. These two genus of the bacteria, some of them that can convert this hesperidin to hesperidin. And actually, we have two INCI names approved international. Okay, one INCI name is for hesperidin, and one is for hesperidin. So what is the INCI? That is a requirement when we develop the anti-aging cosmetic. INCI is the International Nomenclature Cosmetic Ingredients. So we have these two INCI names, and we have technology transfer to a biotech companies. And the biotech company actually they have developed the anti-aging cosmetics. So you probably can search from the Googles. The name is AG Lab. Okay, this means anti-aging. Okay. Oh, I would like to acknowledge my SIS-D2 team members, including the collaborators for the translational medicine and drug development. Okay, I think I'm going to stop here, and I wish everybody have a healthy aging and a longevity. Thank you.
Thank you. So thank you very much, and we have about 2 minutes left. May I ask a question? Do you have a tropical use formula for this your ingredient, your active compound? For yes. Topical use instead of,
the anti-aging cosmetic actually is for topical use.
Right, but all your mouse model is systemic.
Yeah, this is systemic. And actually, I didn't show the data. Systemic is for the whole body, is health. Okay, for the topical treatment, only for the skin.
Okay. Mm-hmm. Another thing is that you have in your liver, seems that there is fatty accumulation, or you want to call it fatty liver. I just wonder, is this cause a problem for systemic use? Is there any hepatic impairment, functional impairment?
Yes, the functional impairment in the liver is due to its age. It's caused by aging. Okay, and because when we deliver the PC19 from the food, and then the first time, so after absorption from the intestine and then the circulation to the livers. So the first line beneficial, what we see is in the livers. So after like a few days, 3 days treatment for the PC, we see the improvement in the fatty livers.
All right, thank you. So
I think hesperidin is already in the market in Japan, so the hesperidin is available. So I think the company is very happy to see the data.
But one thing I want to remind is hesperidin is not a strong SIS-D2 activator. They need to convert into hesperidin. So you need to have a good bacteria in your intestine.
Dr. Rai-Hua Lai. Dr. Rai-Hua is currently the assistant investigator in the National Center for Geriatrics and Welfare Research. Oh, NHI. That was a long name. Sorry. So his talk is entitled Strategies for Mitigating Progression from Mild Cognitive Impairment to Alzheimer's Disease via the ApoE4 cortisol gut microbiota axis. All right. So please, Dr. Lai.
Thank you for the reading. And good afternoon, everybody. I am honored to stand before you and discussing an interesting issue about how ApoE4 promote a specific cortisol mediated mild endocrine atrophy, and also suggest a strategy for reducing the MCI to AD progression through this axis. For the introduction, I think most of you has been known that Alzheimer's disease is a devastating neurodegenerative disorder. AD is the most common form of dementia, counting about 60 to 80% of dementia cases. And currently, there have more than 55 million people have this disease, and due to the population age, this number is projected to triple, reach over 150 million people in 2050. And moreover, the AD is known as the seventh leading cause of the death and a major contributor to disability and dependency among age globally. And this is a serious healthy problem. However, there is currently no cure for AD. Two AD drugs has been approved recently, but however, they could only slow the disease progression, and their effectiveness is very limited. So why is AD so difficult to treat? First reason is AD is a very complicated and heterogeneous disorder with variant clinical manifestation and underlying mechanism. And the second reason is currently treat AD in the AD stage may be too late. So therefore, current approach to AD treatment should be treat AD at the last stage of the disease to focus on the preventive strategy at the early stage. Therefore, the early diagnosis and intervention is necessary. And due to the AD is very complicated and lack of clarity on the molecular mechanism, the precision medicine also should be applied. Among the factors related to AD, I think ApoE4 could be the first step of precision medicine in AD. ApoE is a gene located on the chromosome 19 with the three variants, ApoE2, ApoE3, and E4. And ApoE4 is known as the strongest risk factor for AD. And about 40 to 65% of the AD patient has been known as carry at least one copy of the E4 allele. And it's also known that people carry with one copy of E4 allele could increase the risk to developing AD about 3 to 7 times. And carry with two copies could increase the risk up to 12 times. And since a new approach should be emphasized in the early disease stage, and the specific influence of E4 in MCI is poorly understood, especially in the molecular signaling. So my end in this study is try to identify the key cellular factor in E4 carrier during MCI to AD progression. And here I have using the ADNI data set focus on MCI subject with plasma factor, especially focus on plasma protein, peptide, and hormones, and examine the change of those factors during the MCI-to-AD progression. And our result find that 50 factors are significantly changed during the progression in E4 subject. And interestingly, the result find that those results suggest a cortisol activation signaling, including cortisol itself showed an increased trend in the progression, and also 7 factors are known as cortisol related target, and also 7 factors are known as cortisol related factors. So those results suggest in E4 subject in MCI stage that the predominantly signaling is cortisol signaling, and in contrast to the E4 subject, we find that in the non-E4 subject, the non-E4 subject, there has a different signaling that's predominantly in cytokine and chemokine signaling. Since here we find that since we find that the cortisol activation is a distinguished feature during the MCI-to-AD conversion in E4 subject, so we next are curiously whether cortisol play a specific role in MCI-to-AD progression in E4 MCI subject. And our result find that in E4 high cortisol subject, they showed that a faster AD progression compared to the low cortisol subject and non-E4 subject. And this result find that cortisol play a specific role in E4 MCI subject, and also suggest that cortisol may be as a potential mediator for E4 mediated MCI-to-AD progression. So to test this possible role of the cortisol as a mediator in E4 and MCI progression, we're using the Mandarin randomization analysis. And this is a statistical method that could demonstrate the causal relationship in a factor and observational study. And using the Mandarin randomization analysis, we find that and using E4 as a variant, we find that cortisol could act as a mediator between E4 and AD progression. So those suggest that the cortisol level impacted by E4 may play a role in mediated MCI-to-AD progression. And since we find a causal role of the cortisol in mediated E4 progression, so we are next try to examine whether cortisol has impact on brain atrophy. And our result find that in E4, high cortisol subject has specifically associated with entorhinal atrophy in high cortisol subject compared to the low cortisol subject. And this phenomenon is only found in the E4 subject, but not in the non-E4 subject. And moreover, we find that this brain atrophy is only associated with entorhinal, but not in the hippocampus or the other brain region. So those suggest that E4 with high cortisol level is specifically associated with entorhinal cortex atrophy. And since endocrine play an important role in in receiving the sensory information from the olfactory bud and relaying it to the hippocampus for integrating with many behavior processes. This suggests a possibility that this endocrine atrophy may be associated with some behavior change in E4 MCI subject. So to test this possibility, we have analyzed behavior process in the ADNI cohort in examining the change of those behavior processes in E4 and non-E4 subject with high or low cortisol level. And our result find that the E4 subject with high cortisol level has specifically associated with the eating behavior change. And this behavior change is only in the E4 high cortisol subject, but not in the non-E4 subject. And this suggests that cortisol-associated endocrine atrophy is embodied in the eating behavior in E4 the MCI subjects. So here we find a potential causal association between E4 cortisol and find the role of cortisol in promote E4 MCI progression in this observational study. So we next try to address some mechanistic basis on this study. And two questions has been addressed, first is how E4 modulate cortisol level. For the mechanism, we first try to ask how ApoE4 promote the cortisol level. So we first should we know how cortisol could be regulated in human. And in human, there has two molecular mechanisms could regulate cortisol level. First is through the systematic cortisol production regulated by the HPA axis. And the second is local cortisol activation regulated by the 11-beta-1 HSD1 enzyme. So we next try to test which one is important in E4 cortisol level. And first we focus on HPA axis, and we examine the critical enzyme, critical hormone ATCH. This is a critical hormone in HPA axis. And our result find that plasma ATCH level does not change during the MCI to AD progression. And also we find that plasma ACTH level does not affect the progression of the MCI to AD. So this suggests that the ACTH may not a major contributor to the E4 cortisol level. So how about the local cortisol level? So we will focus on this 11-beta-1 HSD1 enzyme. And interestingly, we find a genetic association of this HSD11-beta-1 SNP is specifically associated with the AD risk in the E4 subject, but not in the non-E4 subject. And this suggests a possibility that this local cortisol enzyme may play a role in the E4 subject. And moreover, we find that the SNP of this gene has associated with the plasma cortisol level, and this association is also in the E4, but not in the non-E4. So those results suggest that the local cortisol activation may be importantly in the E4 and cortisol level. And further supporting this possibility, we find that the HSD11-beta-1 level is higher in the E4 subject compared to non-E4 subject in human endocrine with AD. And also, we find that the similar result in the human hippocampus with AD. So, taken together, those findings suggest that the HSD11 beta 1 mediated local cortisol activation play an important role in E4 MCI progression. But the question still here is how E4 promote cortisol level. From the literature, we find that the E4 could promote the CBP beta activation. And CB beta is known as the transcription factor of the HSD11 beta 1. So those suggest that the ApoE4 may promote the CB beta bind to the HSD11 promoter and activate the HSD11 beta 1 transcription and then activate the cortisol. To test this possibility, we're using a human microglia cell HMC3 to demonstrate this possible signaling. And our result find that E4 could promote the CBP beta bind to the HSD11 beta 1 promoter. And we find that E4 could promote the HSD11 beta 1 transcription and translation. And also, we find that E4 could promote the cortisol level. And more importantly, we find that HSD11 beta 1 inhibitor could suppress this cortisol activation. And take together those results supporting our hypothesis that ApoE4 could promote the cortisol activation through the CBP beta HSD11 beta 1 signaling. And since here we find the answer of how ApoE4 modulates the cortisol level, the next question is how cortisol promote endothelial atrophy of the neural cell death. We believe that the endothelial glucocorticoid may correspond to this cortisol response. In detail, we find that the glucocorticoid receptor level is higher in the endothelial region compared to the other hippocampus region in human. And also, we find a similar result in the mouse. And moreover, we find that the glucocorticoid receptor level is higher in E4 subject compared to the non-E4 subject in human endothelial cortex with AD. So here we find that the endothelial glucocorticoid may play a role in the cortisol response, but how it promotes the neural cell death. From the literature, we find that the cortisol glucocorticoid receptor could promote the VDR level. And also, we find that VDR is known to promote a beta-dependent neural cell death. So here we hypothesize a model that cortisol could promote glucocorticoid receptor bind to VDR promoter and promote VDR labeled and VDR-dependent neural cell death. Supporting to this possibility, we also find a genetic association of the VDR. And we find this genetic association is specifically associated with AD risk in E4 subject, but not in the non-E4 subject. And moreover, we also find that the VDR level is higher in the E4 subject compared to non-E4 subject in human hippocampus with AD. So those results supporting that the VDR may play a role in E4 cortisol pathway and also suggest our hypothesis model is potential. So next, to test this possible model, we using the human neural-like cell line and the mouse neural cells. And we find that cortisol could promote the glucocorticoid receptor bind to the VDR promoter. And moreover, we find that cortisol could promote VDR labeled, and it depends on the glucocorticoid receptor. And moreover, we find that cortisol could promote VDR5 to PV3 and followed in the neural cell death. And supporting our model is possible. So in this part, we find that in the E4 MCI subject, there is a distant pathology relative to cortisol signaling that contribute to the entorhinal atrophy during the MCI2AD progression. And in mechanism, we find that the E4 could promote the local cortisol production through the CBP beta, HSD11 beta 1 signaling, and also we find that the cortisol could promote the neural cell death through the VDR PV3 signaling. So in the next part, we try to know. Here we have the signaling pathway. So the next question is how we apply this signaling for the early intervention. From the literature, we find that the cortisol and the lipid metabolites has crosstalk reciprocal lead. And this suggests a possibility that some lipid metabolites could regulate the cortisol level or cortisol function in humans. So to understand the interaction of the cortisol and the lipid metabolites may help us to find a possible solution. So the next aim is to try to investigate the lipid metabolites involved in the cortisol regulation during the MCI2AD progression. So here I also focus on the NCI subject with the lipid metabolites labeled, especially focus on the short-chain fatty acid, middle-chain fatty acid, and long-chain fatty acid. And examine the change of those factors in the high cortisol subject and the low cortisol subject. And here our result find that the E4 subject with high cortisol level are specifically associated with the lower short-chain fatty acid level. And this association is only found in E4 subject, but not in the non-E4 subject. And moreover, we find that the cortisol level does not associate with middle-chain fatty acid and long-chain fatty acid in E4 subject. And since short-chain fatty acid are main metabolites produced by the gut microbiome, this suggests a possibility that the short-chain fatty acid production bacteria may be changed in E4 subject. So we next try to test this possibility. And we're using gut microbiome data and examine the difference of those gut bacteria change in E4 and non-E4 subject. And our result find that the short-chain fatty acid producing bacteria is lower in the E4 subject compared to non-E4 subject. So taken together, those results suggest that high cortisol level is associated with lower short-chain fatty acid level. And this association may be due to the low short-chain fatty acid producing bacteria in E4 subject. And since we find that short-chain fatty acid is specifically associated with E4 cortisol level, the next question is whether short-chain fatty acid play a role in E4 cortisol-mediated AD progression. And our result find that indeed short-chain fatty acid are associated with associated with higher plasma cortisol level in E4 subject, and also associated with the poor cognitive function, and this is only found in E4 subject, and also associated with CSF phospho-tau level in E4 subject. So those results suggest that E4 cortisol signaling has reciprocally communicated with gut short-chain fatty acid in promoting the MCI2AD progression. And to further supporting this possibility, we find short-chain fatty acid transporter MCT1. This is a short-chain fatty acid transporter that could directly participate in the absorption of short-chain fatty acid in the gut. And interestingly, we find a genetic association of this transporter, and it's specifically associated with AD risk in the E4 subject, but not in the non-E4 subject. And moreover, we find that the risk allele of this gene SNP is specifically associated with lower short-chain fatty acid and higher cortisol level and poor cognitive function in the E4 subject. So, take together those results not only supported existence of the E4 cortisol short-chain fatty acid excess, but also find its role in contribute to the AD risk. Since we find a possible model of the cortisol short-chain fatty acid in gut-brain axis in E4 MCI subject. So the next question is how we applied this signaling. I think dietary fiber may be a possible solution. Since short-chain fatty acids are the main metabolites produced by the gut microbiome through the dietary fiber, and this suggests a possibility that the dietary fiber intake may promote the short-chain fatty acid in a human. Therefore, dietary fiber being made as a possible solution for E4 MCI subject with elevated cortisol level. So we next try to test this possibility using the host database. So here I had using the dietary fiber intake and examine its effect on the cognitive function in host data. And in elder with age more than 65 years old and the MMSE are more than 20 points. And our result find that the dietary fiber intake has a beneficial role in the cognitive function in both E4 and non-E4 subject. And interestingly, when we compare the E4 subject to the non-E4 subject, we find that dietary fiber intake has more dramatically and significantly effect on E4 subject. And this suggests that dietary fiber may have potential to slow down the cognitive decline, especially in the individual with the E4 genotype. So in the last slide, in the conclusion, in here we find that in the E4 MCI subject, the high cortisol level is associated with the early endocrinal atrophy change in the eating behavior and promote a faster AD progression. And secondly, we find that the E4 cortisol signaling is reciprocally crosstalk to gut short-chain fatty acid in promoting MCI to AD progression. And third, in this study, we suggest that dietary fiber may potentially slow down the MCI to AD progression, especially in the individual with E4 by reducing the E4 cortisol signaling. And in the last slide, I will thank my colleague in NHI to have this study, and also thank you for your attention. Thank you.
Okay, thank you, Dr. Lai.
I'd like to introduce the third speaker in this session. The next speaker is Professor Rai-Hua Otsuka. She's the professor of the Department of Epidemiology and Aging in our National Center for Geriatrics and Gerontology. The title of his hot talk will be Healthy Aging and Biomarkers, a result from NILS-LSA focused on fatty acids. So maybe we can hear about similar result with the last speaker about the role of fatty acid in healthy aging. So let's welcome Rai Otsuka.
Thank you. It's my honor to present here. Today, I'd like to talk about the healthy aging and biomarkers results from our cohort NILS-LSA study and focus on fatty acid. Today, I have 4 topics. And first is the introduction of fat intake and fatty acid. Second is the NILS-LSA cohort study. And today, I will focus about fatty acid intake and function and brain volume. And the next is the trend in fat intake and serum fatty acid intake in Japan. And lastly, I would like to discuss about fatty acid for healthy aging. So, nutritional function of fats, there are many nutritional functions of fats. The main is the energy source. And second is the structural component of cell membrane. And fats aids in the absorption of nutrients and increasing bioavailability. And fats derived as smell, taste, and satiety of food. And fatty acid is usually divided saturated fatty acid and unsaturated fatty acid. And today I will focus on the arachidonic acid from N6-PUFA and EPA and DHA from N3-PUFA. So Taiwanese and Japanese diet are so unique diet in the world, and so we usually eat rice and side dishes with egg, chicken, and vegetable and some soup. And this is the energy provided by nutrient intake among older community dwellers. And so we can see this is because based on the publicly data, the energy from Taiwanese is relatively low, but today I would like to focus about the fat energy percentages. So the Taiwanese energy from fat is 29, and Japan is 25.7. And compared to the, they are very lower than the US. US is 33.2. So now in Japan, our energy providing nutrients among Japanese are dramatically changing after World War II. We especially carbohydrates is decreasing after World War II, and protein is also increasing, and fat is rapidly increasing. And over the 2 decades, carbohydrates are also decreasing, and protein is stable, but fat intake are rapidly increasing within the decades. And this is a fatty acid metabolism slide. And so our body could not metabolize linoleic acid and alpha-linolic acid in our body. And we little metabolize from arachidonic acid from linoleic acid and EPA and DHA from alpha-linolic acid. So, and we usually define essential fatty acid, linoleic acid, arachidonic acid, and alpha-linolic acid, EPA and DHA. So this is a representative physiological function of fatty acid. So I'm not specialized about the fatty acid, but there are many kinds of fatty acid physiological function in our body. So today I would like to focus about the brain. So because this is the DHA and arachidonic acid and saturated fatty acid composition in the main organs. So today I would like to focus on brain because our brain has rich DHA. So for example, our body. First, I would like to show about serum fatty acid intake. The main is the saturated fatty acid. Serum and plasma is also equal, but there are low rate of DHA and arachidonic acid in our serum or plasma fatty acid. And similarly, so breast milk is covered in the saturated fatty acid. On the contrary, leg skeletal muscle is abundant in arachidonic acid. But in our brain, so there are much DHA in our brain. So our cells are covered, consist of phospholipids. So in our brain, there are many DHA in the hydrophobic fatty acid. So next, let's move to our cohort study. So NILS-LSA is the National Institute for Longevity Sciences Study of Aging, conducted by National Center of Geriatric Gerontology. And NILS-LSA study started in 1997 and now conducting in 10th survey now. And we followed up 2,300 men and women. They are randomly selected from community dwellers. The baseline age is 40 to 79 years. And we surveyed 2,300 persons every 2 or 3 years and continue to this longitudinal survey. And NILS-LSA have many examinations, especially medical assessments. So today I would like to use some data of fatty acid from blood test and brain volume from head MRI, and 3-day dietary records from nutrition survey and cognitive function. This is the age and sex distribution of NILS-LSA study. So you can see all the study waves have the 2,000 to 300 persons, and in each age decade, each age group, there are almost 280 men and women in each age group, and continue to follow up them now. And data on fatty acids from NILS-LSA divided two parts. The first is a 20-hour fasting blood. So we have serum DHA, EPA, arachidonic acid, and DHLA on the first, second, third wave. In addition, fifth and seventh wave, we have a serum concentration of 24 fatty acids, including DHA, EPA, arachidonic acid. In addition, we measured the 3-day, we assessed the 3-day dietary records, and the nutritional index were calculated, including 45 index of fatty acid. So this slide shows the food and nutrient index by age group. The point of this slide is this slide shows the age group for age group and men and women, their nutrition intake. And the following dietary index change with aging. So fish and EPA and DHA intake increase on the older age group. In contrast to meat and saturated fatty acid decrease in older age groups. This means age-related difference, just the age cohort difference. So it means the older consume more fish and less meat. This is a fat intake and blood levels of DHA, EPA, and arachidonic acid. This is the dietary intake distribution, and we can see the DHA and EPA distribution is skewed to the left side, and the median intake of DHA is 425 milligram per day among our study. Compared to the US men, there are maybe 7 times higher among Japanese. Similarly, EPA intake is skewed to the left side, and maybe the 7 times compared to the US men intake. And arachidonic acid is, it seems to be normal distribution, and median value is 150, and similar to US men. Similarly to the dietary distribution, and serum DHA, EPA, arachidonic acid distribution is almost equal to the dietary intake. So median of serum DHA and EPA much higher than the US. But arachidonic acid, serum arachidonic acid is almost equal US men. So in our cohort, fat intake and serum fatty acid by age group are here. Above is the fat intake and below is the serum fatty acid intake. This bar means different age group. So among the older with the aging, so serum DHA and EPA intake also increasing with aging. But arachidonic acid is only, serum arachidonic acid is only decreasing with men. This slide shows the Pearson's correlation coefficients between serum fatty acid and fatty acid intake. The bar means Pearson's correlation coefficients. The results show the positive association was found in N3 PUFA, especially EPA and DHA. They are highly correlated between the serum fatty acid and fatty acid intake. So next is the fatty acid and cognitive function and brain volume. So I emphasize today's focus is the brain, our brain, because our brain has much DHA. And so our brain, 55% of white matter and 30% of the gray matter's dry weight is lipid. So fatty acids are found in our membrane, cell membrane, and fatty acid makes up phospholipids. And DHA is the composition of DHA among our brain is 11%. There are so high percentage in our brain, and 6% of PUFA in our brain are DHA. This figure shows the fatty acid composition of phospholipids in our brain by age group. So, for example, stearic acid is much contained in frontal gray and hippocampus, but oleic acid is monounsaturated fatty acid is much higher in white matter. And arachidonic acid is almost equal, contains the frontal gray and white matter and hippocampus. And for the DHA, so there are much containing frontal gray and hippocampus, and compared to the white matter. So this slide shows the fatty acid composition of phospholipids in the brain. So this bar means green bar is a control and the yellow bar is Alzheimer's disease, patients with Alzheimer's disease. The composition of arachidonic acid and DHA are decreasing among the Alzheimer's disease. I don't know exactly the age-related or disease-related fatty acid composition changing because these data are derived from biopsy, so we don't know all the age-related difference. But the fact is the difference. So there are aged men or Alzheimer's disease men is decreasing with arachidonic acid and DHA compositions. This is one of the NILS-LSA study results. And at first, I assessed the serum DHA concentration and risk of cognitive decline from 10-year longitudinal cohort study. And so serum DHA was divided into 3 groups, and compared to the lowest serum DHA group, so medium or higher serum DHA group has a lower risk of odds ratio of cognitive decline. And DHA and EPA, they are highly correlated with intake and serum fatty acid intake. So that this result means consumption of blue fish and other fish rich in DHA and EPA reduce the risk of cognitive decline. In addition, so NILS-LSA study to clarify the factor of morphological aging in the brain, we conducted 3-day head MRI data at 6th, 7th, and 9th, and 10th waves. We calculated the 3-day head MRI data from our cohort. And this is one of the results of age-related morphological aging in NILS-LSA study. And this is the age-related change in hippocampal volume. So above figure is total gray matter volume, age-related total gray matter volume loss, and below is hippocampal volume loss. And this slide shows red line is the total gray matter volume and blue bar is hippocampal volume. So in older age, atrophy in hippocampus progresses faster than the total gray matter volume. So this is DHA, EPA, and arachidonic acid intake and the changes in brain volume. So this figure shows that only arachidonic acid were negatively associated with the brain volume loss. And DHA and EPA are no positive association between the brain volume loss. And from the result of this, so we considered the reason of why DHA and EPA are not associated with brain volume. So we focus on the difference of the DHA and EPA intake among Japanese compared to the North America. So in Japan, we have a lot of fish, and the mean DHA and EPA intake in Japan is at least 3 times higher than the other countries. So, and in this study, we conducted sub-analysis focused on the bottom 1/3 subgroups in our study. So this means we focus on the lower DHA and EPA consumption group and conducted the same analysis. And as a result, this slide shows the DHA and EPA intake was negatively associated with the total gray matter volume and temporal cortex volume in this subgroup study. So this is a summary of the results. So our cohort study, the lowest group, DHA group, is a high risk of brain volume loss, and second risk is the second group. And similarly, EPA lowest group is a higher risk of brain volume loss. And for the arachidonic acid, the bottom of the 1/3 group is a higher risk of compared to the other group. This means the lowest DHA or EPA intake and lower arachidonic acid intake were associated with cognitive decline and brain atrophy in Japanese population. In other words, rarely eat fish or low animal food consumption is a risk of cognitive decline and brain atrophy in Japanese population. And then let's move to the next trends in fat intake and serum fatty acid intake in Japan. So Japanese fat energy percent is dramatically changing after World War II. And in the background, our meat and fish intake among Japanese were dramatically changing after the World War II. The top of the animal protein products from meat is changing, and so meat is increasing, and fish and shellfish were decreasing. Almost 2008 or 2010, the top of the animal foods were changing among Japanese. And similarly, saturated fatty acid and N6 fatty acid are also increasing. N3 buffer among Japanese over the 2 decades are stably maintained. But N6 PUHA and especially saturated fatty acid intake among Japanese are increasing. So this slide shows the serum arachidonic acid, EPA, and DHA concentration in our NILS-LSA cohort study. This is a 12-year longitudinal data. And 12-year longitudinal data shows serum arachidonic acid concentration increase in all sex and age groups. And when we focus on the fatty acid composition, so we only calculated 2 study waves for fatty acid composition. So total fatty acid were increasing in the 40s men and 40 and 60s men, women, and this is the fatty acid composition. Only monounsaturated fatty acids are increasing in 40s in men and 40 and 50s in women. And arachidonic acid composition were increased both in men and women in all age groups. And when we adjusted for the corresponding fatty acid intake, serum fatty acid composition, arachidonic acid composition increased in men and women in their 40s, 50s, 60s. This means our serum arachidonic acid composition were increasingly independently of fatty acid intake. So recently, Hisayama cohort study, this is one of the representative cohort study in Japan, also reported the same results. They reported the serum EPA increasing from 2002 to 2012. And contrary, the arachidonic acid were increasing, EPA is decreasing in some age group, and arachidonic acids are increasing in all age groups. And they focus on the EPA per arachidonic acid ratio, and serum EPA per arachidonic acid ratio decreased within 10 years, especially in middle-aged Japanese individuals. So EPA and arachidonic acid ratio means lower risk of atherosclerosis. So, for example, Canadian ethnicity Inuit has a high EPA arachidonic ratio, and Japanese is almost 0.2 or 0.3, and US is almost 0.1. And so the ratio is decreasing among the Japanese. So finally, I would like to discuss about fatty acid for healthy aging. So in Japanese dietary guidelines recommend 20 to 30% from fat energy is recommended. So today I didn't mention about the fatty acid and muscle health and the prevention of frailty, but our cohort study reported that saturated fatty acid and fat intake associated protective for muscle health and the prevention of frailty. And I think approximately 20 to 30% of energy of dietary reference index among Japanese might be reasonable in Japanese. But we already are getting that percent. And today I did not mention about the saturated fatty acid. So this slide shows the saturated fatty acid intake and the incidence and mortality rates of stroke, and lowest saturated fatty acid intake is the risk of stroke. And now our study is almost saturated fatty acid intake is almost 7% in men, and women 7.9% from saturated fatty acid. And so this line is a US cohort, and the US cohort is they consume much saturated fatty acid intake, but the Japanese consume the lower rate of saturated fatty acid intake. And I think almost 7% energy intake based on the dietary reference index among Japanese, it might be reasonable for Japanese individuals. So again, I would like to say to avoid a decline in DHA or EPA intake, fish should be consumed more than once or twice a week. So to avoid the lowest DHA and EPA intake. And to avoid a decline in arachidonic intake, especially in the old people, moderate amount of animal animal-based food such as meat, fish, eggs should be consumed. So I have two suggestions. So to eat DHA and EPA intake effectively, so I would like to show DHA and EPA residuals by cooking methods. So for example, we usually eat Pacific sori. So when we eat sashimi, Pacific sori as sashimi, we can get DHA and EPA almost 100%. But when we grilled 10 or 20% loss, and frying is almost the same. And deep fry is maybe 40 or 50% loss of by cooking method. So it is avoidable to eat deep fry when we eat DHA and EPA more. And to increase the DHA and DHA and EPA levels, so one of the strategies to eat alpha-linoleic acid. Only 5% of alpha-linoleic acid converted the EPA, and EPA to DHA is only 0.5%, but we can create the EPA and DHA from alpha-linoleic acid in our body. So this is the last slide. So today I focus on the fatty acid intake, and our data shows the total fatty acid for Japanese is maintaining an energy level less than 30% is better, and saturated fatty acid is to keep almost 7% energy, and DHA and EPA to eat fish over 1 or 2 times per week is better, and arachidonic acid to eat moderate animal foods. And it is important to check the balance of N6 and N3 PUFA. So it means that to eat both fish and meat is better for the Japanese. Thank you for your kind attention.
I'd like to introduce the last speaker in the session, Dr. Kun-Pei Lin from National Taiwan University Hospital. The title of his talk is Analysis of Plasma MicroRNAs as Biomarkers of Dynapenia in Older Adult. So let's welcome Dr. Lin.
Thank you, Professor Lin and Professor Alai. It is my honor to be here and present this topic. Our study is the analysis of plasma microRNAs as a biomarker for Dynapenia in older adults. Dynapenia, most people know sarcopenia, but Dynapenia is very important too. We will introduce the Dynapenia according the phenomenon and the definition and the characteristic of the Dynapenia. And because Dynapenia and the sarcopenia are very similar, and they share the same symptoms and the same signs, so we will introduce the distinction between these two diseases. And the third, the impact of the Dynapenia on overall health and the functional abilities in older adults. Dynapenia, the definition of Dynapenia is the age-associated loss of muscle strength, and the predominance caused by non-specific conditions. In other words, if patients with specific disease such as the stroke or muscle damage, neural damage, we can say they may be caused by the secondary factor or secondary cause. And the characteristics of Dynapenia is the same or similar to the sarcopenia. It has a slowness, weakness, and a low physical performance. However, there is currently no agrees upon the algorithm to diagnose Dynapenia. These two charts illustrate how Dynapenia is influenced by the age. In the left chart, the red lines represent the muscle mass, and the blue lines represent the muscle strength. We can see the red line, the muscle mass is declined later than the blue lines. So the muscle strength decline is occurred early than the muscle mass. As the age, the right pictures is show the data is from the South America. We can see in different age group, the prevalence of Dynapenia is also different. In the people with 50s, the prevalence of Dynapenia is about 17%. When the people consider they are older, if they in 70s, the Dynapenia prevalence becomes approximately 30%. So this indicates the Dynapenia prevalence is higher than all adults. This slide shows the distinction between the Dynapenia and the sarcopenia. The main difference between Dynapenia and the sarcopenia lies in muscle mass. Dynapenia typically refer to a functional decline, and this means the blue area. The blue area shows a decreased muscle strength, and the gray area that shows a decrease in muscle mass. Their combined zones, the dark gray area, is the sarcopenia. So we can say sarcopenia is equal to the decreased muscle strength, Dynapenia, and the decreased muscle mass. However, the age loss of muscle strength is weakly associated with the loss of muscle mass. For example, if the patient with weight gain also had muscle strength decrease, but their muscle mass still increase. So, when our focus is the muscle strength or muscle functions, consider the sarcopenia may be not specific. Dynapenia may be the more, when we focus on the muscle function or focus on the muscle strength, Dynapenia may be a better target. Dynapenia also has impacts for overall health. First, it will increase the risk of physical disability and poor physical performance. The people with Dynapenia also increase risk of falls. Maybe they have weakness or slowness or bad or worse environments that they increase the force. And they also have more risk of impaired metabolic health, such as hypertension, diabetes, or hyperlipidemia. Because muscle and bone have a very close relationship, so the patient with Dynapenia also has higher risk of low bone density. Some studies also indicate that Dynapenia has correlations with cognition function. And because the Dynapenia makes people's weakness, so they also reduce quality of life. And overall, we had found the Dynapenia had increased mortality in the elderly. Several biomarkers in Dynapenia have been described. The most described biomarker is the MicroRNA, specific on the muscle specific MicroRNA. And some inflammation markers had been demonstrated in previous studies, such as CRP or the ESR. Some proteins like insulin-like growth factor or myostatin has also been found. Cortisol and testosterone, these endocrine markers, also be found in some of the Dynapenia cases. However, most biomarkers are uncertain, and ongoing research may identify additional markers to refine the significance of existing ones. This slide shows the microRNAs, how the microRNAs functions in our physiology. The message RNA produced by the typical gene and proceeds the synthesis of protein. However, microRNA acts on the message RNA and leading to the degradation of the target. That will inhibit the protein synthesis or then block the protein assembly. This slide shows the postulated mechanisms pertaining to microRNA function and the Dynapenia. When people become aging, there are physical changes that occur at the cellular level, such as mitochondrial dysfunction, chronic inflammation, metabolic dysfunction, cell apoptosis, and insulin resistance increase. They were found associated with MicroRNAs regulation. These factors may lead to the Dynapenia. And patients with Dynapenia also lead to the physical inactivity, and they may have insufficient nutrition. Both of these two conditions may worsen the physiology change. So it became a vicious cycle. So the MicroRNAs plays a certain role in the physiological change. So this is our studies methodology. We will select the patients from our OPD, and the demographic information will be collected. Then the plasma samples will be collected and processed. The identification and the quantifications of specific microRNA will be done. This is our study flow chart. We enroll patients aged older than 65 years old and who visit our geriatric OPD. Some will be excluded, including the unwilling or the total dependence like Spiriton status, or the cancer patient or disease terminal case, because their function or their prognosis was different from the community ones, and who are incapable to be evaluated, because we need some evaluate tools. So if they have total hearing impairment or vision impairments, they cannot proceed this evaluation. The sixth, patient with stroke or neurodegeneration disease will be excluded because, as I mentioned above, patient with specific disease leads to the muscle weakness or neural damage. They may consider as a secondary effect, not the pure Dynapenia. And these patients will be tested physical and functions performance, and measure the body composition and collect the biomarker, including the microRNA. We will do the age and sex match compare. And finally, we found the normal group about the 38%, and the Dynapenia group is about the 34%. They all for the prepared sample and for the microRNA analysis. Our studies criteria for Dynapenia definition is for the two criteria. Criteria one is the grip strength. In men, if they had less than 28 kilometers. In women, if she had less than 18 kilometers, was considered a lower grip strength. As for the walk speed, we use the 6 meters walk test. If they were tested lower than 1 meters per second, they were considered as a low walk speed. If patients meet the criteria 1 or 2, will consider the Dynapenia. These criteria were set based on the Asian working group for sarcopenia. When they correct the plasma, the chromosome for microRNAs, we will check the total RNAs reaction and the small RNAs library preparation. And then the size collection, and we will do the most important step, the next generation sequencing. After the next generation sequencing, we will perform scrutiny and read the filtering. Adaptive remove filtering against other RNA species and annotations against human reference genomes. And at final, may find the novel microRNA discovery. This is our sample process summary. Also, we had 72 participant samples curated, but there were 2 samples because of poor RNA quality was excluded, and there was 9 samples because of poor sequencing quality was excluded. So as the final, they had a total of 61 samples was collected for the downstream analysis. The red table is the simple demographics analysis. Because we had age and sex match as the first, but so the age and the sex statistically was showed not significant. We also performed other disease or other functional evaluations, but not shown in this table. The other disease like the diabetes or hyperlipidemia, hypertension was not significant in our study. Why we chose the next generation sequencing? It has 3 advantages. First is high throughput. NGS allows the simultaneous analysis for hundreds to millions of sequences, providing high throughput data generation and compatible for the comprehensive MicroRNAs coding. 2, it has a very high sensitivity. NGS can detect low abundance microRNA, offering high sensitivity for the discovery of rare or low expressed microRNA. So if the microRNA was at the very low level and the distribution to very right, we can find it. And 3, NGS provides a comprehensive analysis of microRNA, not limited to the species known microRNA. We don't need to review hundreds of papers and to find previous studies chosen RNA. We perform a total complete microRNA analysis. That will enable the discovery of normal microRNAs and assess of overall microRNAs expression patterns. This is our result. The principal component analysis reveals plasma microRNAs difference between the Dynapenia and the control samples. In this principal component analysis, we can see the distribution of the 2 principal components, PC1 and PC2. There are still some difference in the distribution of the blue dots. Blue dots represent the control group, and the pink dots represent the case group. So these 2 groups has significant difference distribution. These pictures show the differential expressed microRNAs between Dynapenia and the control samples. The left table is volcano plot. The volcano plot highlights statistically significant difference in the microRNA. The x-axis represents the expression difference of microRNA. The further to the left or the further to the right means the greater of the difference. While the y-axis represents the statistically significance. Typically, if there were more upper zone, they have more significance in the study. So when we show, so we can observe the blue dot as upper left. It means the negative correlation to the Dynapenia. And the right and the pink dot in the right upper zone, it shows a positive correlation to the Dynapenia. And the right picture is the heat map. Everyone knows heat map is a very good visual presentation for the ORNA's expression. We can find the blue is the control group and the case group is the pink. The lower expressions is the blue, purple area, and the red areas means is the higher expression. We can see the two areas had separate clearly. We employed the 10 microRNAs in predicting Dynapenia. It's focusing on the selective positive features. The heat map on the left illustration demonstrates their distributions. Meanwhile, the right chart shows AUC curve, shows AUC area for the predicting Dynapenia and reaching a promising value, 0.77. We use another 13 microRNAs in predicting the Dynapenia, focusing on the selecting the negative features. The heat map on the left illustrates their better distribution. We can see the purple, blue, and the red area separate very clearly. Meanwhile, the right chart shows the AUC for the predicting Dynapenia, reaching the SM values 0.82. So the combination of supposition and negative features for prediction. We combine the positive and the negative model and combine them to a composition model. The AUC was about the 0.81. This is our future direction. In these studies, we identified a total of 10 microRNAs possibly and the 13 microRNAs negatively correlate to the Dynapenia. And this microRNAs will be composition to a compound of microRNAs model. We would use them maybe to develop a validation model in the future or the verifications through the in vitro microRNAs mimic transaction. This is our conclusion and the future direction. We're using the NGS for microRNAs research. We suppose the NGS is a very good tool, especially in the complex or multifactorial disease, such as Dynapenia, sarcopenia, or frailty. They can provide a deeper understanding to this condition or disease. And our studies identifies approximately the 23 microRNAs associated with Dynapenia, and observing they has a classical effects in certain specific regulation mechanism. This may provide additional reference data for future research on Dynapenia, sarcopenia. The last, currently because there is no clear diagnosis or treatment or management guideline for Dynapenia, and we want everyone to get more attention to this condition because sarcopenia, as I described above, the muscle mass will decrease later, become the muscle strength decline is early than the muscle mass loss. So Dynapenia will be found earlier than sarcopenia. So if we had more tools to detect or screen the Dynapenia, it may prevent these patients become to the sarcopenia. So our findings maybe give us screen tools in the future. Okay. This is the future directions later may be performed. The identified microRNA and their sequence in vitro synthesis of microRNA maybe we give the mimics or the inhibitor, and the transaction to the microRNA to the in vitro cells. We want to maybe the muscle cells is the more suitable because Dynapenia or sarcopenia may occur more specifically in the muscle cell. And the screen shows difference in the expression the pro-inflammation cytokines, like the interleukin-6 and TNF-alpha. Factors in protein gene degradation is also interesting mechanism. The factors in mitosis pathways may be the next topic. And then we will reconfirm the results by MicroRNA inhibitor in vital cell model. So this is my speak. Thank you for everyone.
If you have any questions, feel free raise your hands. Oh, Gustavo?
I got a question for Dr. Otsuka. Is there an evidence that or there is a high intake of saturated fatty acids? Is there an evidence that there is a change in the composition of the fat in the muscle in terms of becoming more saturated?
Okay, thank you. So we have only 2 times fatty acid composition, and saturated fatty acid composition is maybe not so changed. And so we only have a serum fatty acid composition. We have no muscle fatty acid composition. I don't.
We need biopsy to see the fatty acid composition in the muscle, so it's very difficult for us. Any other questions? Oh, please go ahead.
I have a question for Professor Tsai. Thanks for wonderful and beautiful talk. My question is about that. We talk about that genomic instability is associated to the aging and aging process. My question is about that. Several people they created a drug for DNA repair because they talk about that it can because it promote the DNA repair ability, so it can restore the genomic instability. So my problem is that does the CIS2 has a similar function to DNA repair?
The short answer is I don't know. Okay, I haven't tried, but I can take a guess. Okay, and because the 62 activators actually there was help maintain all the structure and function, almost all the tissue we examined. So I think at least that improve the mitochondrial functions and also decrease the oxidative stress. And because the genomic instability, most of the problem is caused by the oxidative stress and the free radicals. So I would take a guess, but I haven't tried in the mouse. That would decrease the, the genome instabilities. And so the result is, the genome is become more the integrities will be increased. But welcome to join the studies.
Thank you. And I also have another question for Professor Otsuka. Thanks for your interesting and nice talk. Okay, I would like to share our study. Previously, we used the lipidomics to identify the breast milk. And our data is very interesting is that we found that the EPA and the DHA are not related to neural behavioral effect in infants. So it means if you eat a lot of the fish oil, maybe not important for the infant neural behavioral development. And so I have no idea if you are, but we look for the information is that the EPA and the DHA, the function of EPA and the DHA are to can block the oxidative stress. So that's we found not for the neural behavior. My question is that the quicker question is that do you check the free fatty acid, free form of the fatty acid in your study, or just the total form of the fatty acid?
Sorry, we have no free fatty acid, just a total fatty acid in serum.
Thank you.
Any other question? Oh, please.
I'm Wei Jingchen from National Health Research Institute. I have a question to ask Dr. Kun-Pei. I think your study is very interesting because you're looking from the microRNA. But I have two general questions. The first question, based on your result, it seems to me very not easy to explain because when you combine positive correlate microRNA versus the negative correlate, but the total AUC almost the same. So maybe it's not a good way to take advantage of the microRNA because usually one microRNA regulate more than probably 100 genes. So because now you are based on cross-sectional microRNA, so at the same time, so many different factors can contribute. So one possibility is because you have a clinical data, right? So for example, you have data 6 months data and also measure their muscle strength. So you can compare whether from one state to the next state and some microRNA consistently overexpress or downregulate, and maybe then you have more confidence. Those overexpression or downregulation was really associated with Dynapenia. So that's one question and suggestion. The second one is in your official direction, you are trying to use some cell line, right? But for that particular approach, it's also very difficult because you saw many different genes under regulation. So another possibility is if we have 2 times of microRNA, and then using the 2 times blood, you're using the RNA sequence. And then when you search for microRNA, you only search for those with a very large fold change, okay? And then try to look at the gene presumably regular by at least a couple of microRNA, and then examine the RNA sequence to see those gene expression. So that probably can help you to identify some gene that can be explained why they are involved in the Dynapenia. But for microRNA, it's very difficult to explain because it covers too many genes. So that's my suggestion.
Thank you, Professor Chen's suggestion. And the time, when the times go through, the patients will become different conditions. So we also had adapted a cohort. So they will be received second times to the microRNAs is the change, and this is will in our further plan. So maybe the microRNAs change or microRNAs the library will be compared to the previous or the later. We have set the 1 year later to check this the change of including the muscle strength or the microRNAs change. We'll set the 1 year later follow up. And thank you for your suggestions for this future direction. And the second is muscle is the cells. We didn't know this microRNAs regulation more and more mechanisms in the cellular types, but we just want to set a simple environment to observe is the change. But indeed, the total change of the Dynapenias cannot see in the just in the particular specific cell type. Maybe we can observe the clinical condition in the human or in animal model, not just to transaction to the cell type. Thank you.
Yes, sir.
I have a connected question to Dr. Lin about the microRNAs. Where do these microRNAs come from? Because how do you know that these microRNAs come from the muscle, for instance? Because in the muscle, it's interesting, the microRNAs are usually packed in extracellular vesicles. So they don't usually soluble, the microRNAs that come from the muscle. So how do you know, first, where do you think your microRNAs of interest come from? And second, are you really measuring, or are you planning to measure them, or are you including in your measurements the microRNAs that are already packed as extracellular vesicles?
Thank you. The sample is from the plasma serum. It's not for the muscle. Muscle is our future, maybe want to transfection, but in our study, the microRNA sample is from the serum, the plasma. And this plasma maybe the total serum is not in because we had considered the exosome first, but we also choose the total serum to analyze the microRNA. So the muscle cell is not in our study.
Yeah, thank you for all of our speakers. I enjoy very much for today's speeches. And I think three of our speakers talking about the nutrition. Professor Tsai suggests that we may have to eat more peels of Ting-Fen to increase our healthy longevity. And Professor Rai talking about the dietary fibers can increase or preserve our cognitive function. And Professor Otsuka said that the EPA, DHA related to fish intake, maybe it can increase our healthy longevity. So I want to ask all of you, make a comment for the ordinary people, especially for the older people. What is the food composition they have to take? The orange peel or dietary fiber or fish? What is more important? Thank you.
I have been asking about how much of the pills is enough to promote the longevity. The answer is it's too difficult because you need tons of pills each day. So actually we are developing the healthy food or the food and to extract the hesperity from the orange peels. And then because the orange peel, the hesperity is not actually a strong SIS-T2 enhancers. So we are now trying to using the bacteria in vitro to convert the hesperity into hesperity and to develop it into a health food. So I think it's probably coming soon next year. And through that way, that probably is more feasible to promote longevity. But only eating the pills, I think that is good for the intake of a little bit of hesperidin and probably is the fibers. But for the instant effect on the anti-aging, I think that you need to wait for the food that contain the hesperity. Yeah, that's my comment.
For my opinion, I think first I think it depends on what genotype you have. Because I propose that dietary fiber could have effect on cognitive function, especially on E4 subject. And on my other study on the non-E4 subject, I think there has some other nutrition is important on the non-E4 subject. For example, the antioxidant is very important for the non-E4 subject. And in the journal, I think the anti-inflammatory or antioxidant is very commonly in the aging process and the dementia process. Maybe those nutrition is commonly can be used in most subject of the with age of dementia. This is my opinion. Thank you.
So in my opinion, so today I showed the result of fatty acid, but my opinion is rather than the nutrition, so I think the psychological well-being in diet is a more stronger effect for older adults' health. So in our data, so satiety of food or delicious to thinking about food is a predictive factor to protect the appetite, to keep the appetite. So I think that for the elderly, so eating fish and peel and the other thing is important, but I think the psychological well-being is more better older. So, but today I didn't mention about it.
Basically, we do not recommend a specific nutrient. We stress the importance of dietary diversity, including fish, meat, dairy product, and so forth. So, well, if we have a lot of evidence that show that,
Go ahead, please.
Specific nutrient is important for muscle health, brain health, cardiovascular health, but it's, we need to be cautious about the interpretation of the data. Go on.
Changwon from Korea. I have a short question to Otsuka. So I heard that when you measure fatty acid, the fatty acid level of red blood cell is much more stable than blood level. So did you consider the RBC level of fatty acid rather than plasma level?
So we have not red blood cell fatty acid.
Yeah, you measure the plasma blood level of fatty acid level, right? But instead of that, red blood cell fatty acid level in RBC, red blood cell is much more stable and more representative. I heard that. So did you consider that?
No, we have not.
Just as a question, as I'm asking. Thank you so much.
I'd just like to ask Professor Otsuka a very interesting question is, now more and more people adopt vegetarian style. So they think vegetarian is good for health. So in your opinion, is any way there can be a very balanced vegetarian style that can still lead to a long, healthy aging process? Because you asking people to eat, at least one time per week fish, and also to balance the fish and the animal food. But for vegetarian, they don't eat fish. They don't eat animal fat. Then, if they ask you, then what can I do to have a very healthy aging? Do you have any suggestion for that?
Vegetarian.
Vegetarian.
Sorry, I have no idea, but animal protein is important for health, and so for vegetarian, they don't eat fish, and their blood levels of EPA are very low, and so for them, I would like to advise to eat alpha-linoleic acid to create their body to EPA and DHA, and so.
Vegetarian I think it's better for health in young people. The only problem with the vitamin D deficiency, so we need to have a supplement of vitamin D for vegetarian. But in older people, I think we need animal protein for longevity. That's my belief, but I don't know about. Okay, please go ahead.
Thank you for your wonderful presentation. I have a question to Professor Otsuka. When we think about Japan situation, like let's say compare 1950 to the nowadays, the longevity and healthy longevity is increasing. But in terms of food consumption, we are increasing meat consumption, not for the fish consumption. I think it's kind of paradoxical, the evidence that you presented. do you have any idea what? Or I'm not sure what we should increase.
Yeah, thank you. So so you said so our fish consumption decreased and meat intake is increased, and our healthy life longevity is increasing. So I think that animal protein is important for Japanese because we had a low animal protein and fat intake previously, but now our consumption of animal protein is almost good for our health. But at the top of the food stuff from in the animal protein is changing. Our DH and EPA intake are decreasing.
So you may agree that the carbohydrates should decrease to increase the animal protein, right?
Maybe it affects our longer longevity.
Yeah. Maybe that's the consensus.
I think it's multifactorial to explain healthy aging in Japan or other countries. For example, national health coverage is very important for healthy longevity, and also longer education and less smoking. And so those, in combine those factors, our healthy longevity and also life expectancy is increasing in the last 50 years. So maybe nutrition is one of them, but it's hard to address which is better or which is worse. So, but generally speaking, we need good fat to maintain a sufficient amount of energy and good protein and good fat in addition to carbohydrate. That's my belief. Do you have any opinion?
I agree with you.
Any other question? I have a question to Professor Tsai. So you have a good reporter system. So have you tried whether hespertin can increase the expression of reporter gene in your mouse? Which organ is more expression of cystic gene is enhanced by administration of hespertin?
I think all of them are activated by cystic-2, but yeah, almost all the tissue are exempt. But because of the basal level of cystic-2 in different organs, they are different. So and also under different situations. For examples, I show one slide. If you have a calorie restriction or starvation, the most important to increase the highest organ is the liver. Okay, and next is the muscle. But when we treated the hespertin, hespertin will increase almost every tissues and increase to the levels and then all compatible to their young age. So it's quite amazing. But if I use a different cystic-2 activator, because we got a panel of cystic-2 activator, and some of them they are small compound, and then we aim to develop is the clinical drug. And then particular with different series of novel compounds, and actually the target is different. They have different mechanism of action. So, but in general, if the hespertin, in terms of hespertin, I think it's almost all the tissues are exempt. They increase the cystic-2 to the levels that are compatible to the young age. Okay.
I have a related question that at the beginning, why you chose the transcription regulation instead of just the cystic-2 activator?
No, actually the regulation of the cystic-2 is at multiple layers. Not only the transcription and also the RNA stability and the protein stabilities. Almost every layers they have different regulations. I choose the at initial stage I choose the transcription because it's much easier to get into. It's just because it's much easier to set up the platform.
As I mentioned, hespertin is available as a supplement in Japan and maybe in Taiwan. So
talking about the hespertin.
Hespertin, yes. So have you identified the good microbiota to convert hesperidine to hesperidine?
Oh, that is the top secret. Yeah, it is a top secret. And I just mentioned that, that discuss with our president how to have a patent to protect. Yeah, it is a top secret.
I see.
No, I'm not to tell you.
Okay, it's a top secret. Thank you. Okay, so we are running out of time. So if there is no other question, I'd like to close the session. So please give the big applause to all the speakers. Thank you.
All the speakers, please come to the front of the stage. The moderators will present special gifts to thank for your contributions for this session.